Hey, good morning. My name is Kyle Schaefer, and we're going to look at the processes of glycolysis and glucogenesis with a focus on aldolase. Okay, so aldolase is going to be the key step, or one of the key steps in both processes, that is going to catalyze the reaction from fructose 1,6-biphosphate and splitting that 6-carbon molecule into 2,3-carbon molecules, glyceraldehyde 3-phosphate, and dihydroxyl acetone phosphates. Okay, so here we go. Let's do it. Okay, so just a brief overview. Okay, so glycolysis. We're more familiar with glycolysis, breakdown of sugars. Okay, so an anaerobic respiration is where we're getting our quick energy. Aerobic respiration, glycolysis is the first step to break essentially glucose into pyruvate. Okay, so taking a 6-carbon molecule and breaking it down into 3-carbon molecules, and those will go to the mitochondria for complete breakdown, and that's how we produce all of our energy through oxidative phosphorylation. Okay, so glycolysis, we're breaking it down, 6-carbon into 3 carbons. Okay, glucogenesis, on the other hand, is deriving glucose from non-carbohydrate sources. So anytime we have low blood sugar, okay, being able to break down fatty acids and glycerol and converting them into glucose that can be used for energy sources. So basically, glucogenesis, we're going to raise the blood sugar, okay, and glycolysis, we're going to break down that sugar into energy. Okay. All right, so not exactly opposites, but they're going to be close, many similar steps. Okay, so the fact that they're reversible is a key issue here. So, all right, so here we go. So liver, we're going to get more glucogenesis. We get glycolysis there as well. We get breakdown. Okay, but liver stores things like fats and fructose and all kinds of things. So we're going to break that down or reconvert it into glucose. So if you think about, okay, what happens in anaerobic respiration? Okay, glycolysis is going to get us that quick energy. But what's the end product? The end product is lactic acid. Okay, lactic acid will convert to lactate, and then that lactate can be reconverted by adding a little bit of energy back into glucose. Okay, so that's glucogenesis. We're converting lactate, we're converting glycerol, we're converting fructose into glucose. Okay, and again, raising blood sugar so we can produce energy. Okay, so when is our energy low? Well, fasting, those kind of things. Anytime we're without food for a long period of time. Intense exercise, we're burning too much energy. Okay, could cause glucogenesis or low blood sugar. Diseases, obviously diabetes and things like that, are going to play roles in blood sugar. Okay, glycolysis, obviously we see more often, primarily in the muscles. Also, we'll look at the brain a little bit. Okay, we're going to break down glucose into glycogen. Okay, well, we're going to break it down to pyruvate, let's face it. Most of the time we're going to be doing oxidative phosphorylation. Okay, but again, the glycolysis process we're going to focus on is just the first step, which would break it from glucose into pyruvate. Okay, so what are the major steps of glycolysis? Okay, hopefully you've been through this before with cellular respiration. Okay, so we're going to have, sometimes broken into 2 groups of steps, sometimes into 3. Okay, we're going to have the energy input phase. We're going to have that splitting of glucose, which is where we're going to use aldolase. Okay, that's where the enzyme aldolase is going to come in as step 2 here on the splitting of glucose. Okay, and then we're going to have the energy release, or we're going to get energy out of this process, and that's the whole thing. Okay, so we'll look at the actual steps here in a second. Okay, but the big thing is to remember what are the keys. What is the key of glycolysis? And that is we want energy. Okay, so glycolysis will produce a small amount of quick energy in the form of 4 ATP. Okay, the energy input step though does cost us 2, so we're only netting 2 ATP here. We do get some NADH, which is an electron carrier that we'll use to reconvert pyruvate into lactate if we do not go through oxidative phosphorylation. Okay, otherwise it would be used in the electron transport chain. Okay, And then of course, our carbons are going to end up as pyruvate. So here we go. All right, step by step. Again, we're going to start with a 6-carbon molecule. Okay, let's go red here. So glucose, we're going to have 6 carbons here. Boom, 6 carbon. Okay, we're going to watch that carbon break down. So we're going to go through multiple steps here. Go through every step has an enzyme. Okay, so glucose into glucose 6-phosphate. This is going to be our energy input step. So notice we got to use ATP here twice in these first 3 steps, okay, to essentially activate the molecule. Okay, glucose is a sugar; it's not going to blow up on its own. Okay, we need to activate to make it chemically active. Okay, and that's where ATP comes in, adding some phosphate groups that are going to change the structure of the molecule, which is going to get us to that split state. Okay, so here's our split state. Okay, we got 1,6-biphosphate. Okay, and we're going to split that into 2 3-carbon molecules. So this is the big one. We go from 6 carbons up here with fructose. Okay, and we're going to break that into 2 3-carbon molecules here, glyceraldehyde 3-phosphate and dihydroxyacetone phosphates. Okay, which actually hydroxyacetone can be converted into glyceraldehyde 3-phosphate, okay, as you see there. Okay, and then we're going to get our energy release step. Okay, we're going to make some ATP here. So remember, we're going to go through this twice because we got 2 3-carbon molecules. So we get that 2 ATP steps times 2 will give us our 4 ATP. Okay, we also get our NADH. Okay, and our end product is a 3-carbon molecule here in pyruvate. Okay, so key steps for glycolysis. Again, we're going to input energy here on the top. Okay, we're going to split that 6-carbon molecule into 2 3-carbon molecules. And the last stages, we're going to get that phosphate back and create some ATP. So we basically double our input. We put in 2, we get out 4. Okay, so think of it like doubling your money. Okay, not a bad result. Okay, so this is glycolysis again. First step in oxidative phosphorylation. All this pyruvate and the electron carriers would go to the mitochondria for further breakdown and energy production. Without oxygen, anaerobic, okay, quick energy, glycolysis is going to produce that little bit of ATP, and then this pyruvate will be converted into lactate or lactic acid, okay, which will be sent to the liver and reconverted into glucose through glucogenesis. Okay, same thing. Okay, breakdown of glucose. Okay, not quite as detailed, but same general principle. All right, so I guess before we end glycolysis, okay, glycolysis again is breakdown of sugar. So insulin is going to cause us to take up more sugar. Glucagon is the hormone is going to cause us to take up less. Okay, so depending on that, understand glucagon and insulin are going to control both processes. So when we get to glucogenesis right now, okay, glucogenesis, glucagon is going to turn it on. So the enzyme, or excuse me, the hormone, glucagon released by pancreas, okay, is going to increase glucogenesis in the liver, which is conversion of non-carbohydrates into glucose. So it's going to attack our fat storages. It's going to reconvert lactate. Also, some amino acids such as alanine can be converted. Okay. Again, primarily happening in the liver. It can happen in the kidneys. Can also affect the small intestine, actually. Okay. But primarily we're looking at the liver. Okay. So what would cause this? Obviously low blood sugar. Well, think about what would happen with low blood sugar. Okay. low-carb diets. Okay, things like the Atkins diets where we're lowering our carbs to directly cause our body to attack fat storages. Fasting, okay, whether it's just you're going without food for a period of time, again, dietary, could be religious, okay, whatever the case may be. Usually this is going to activate 8 to 12 hours without food, okay. High activity, okay. And the last one is, it's also part of your circadian rhythm, okay. So think about it, we're going to be more or less active based on time of day, sleep, things like that. So glucagon and insulin are both going to play a role in activating and keeping our blood sugar even while we're asleep. Okay? So for glucogenesis, glucagon is going to turn it on, insulin is going to turn it off. Okay? The other enzyme that, or the other hormone that affects is going to be cortisol. Okay? Out of the adrenal glands, and it's going to activate glucogenesis during fasting. Okay. The other controls are high levels of AMP and fructose 2,6-biphosphate are extreme inhibitors of glucogenesis. Okay, because that's indicating that our sugar levels are appropriate and therefore we don't need to reconvert. Okay, so glucogenesis, mainly blood sugar low, mainly in the liver. Okay, and again, many steps in glycolysis are reversible. Okay, so it's basically going to be the same process going backwards. Okay, so if we look at glucogenesis, right, let's see, what's our best view? Probably here. We'll look at this one. Okay, so glucogenesis, as you can see, it's almost opposite of glycolysis. Okay, so what did we produce in glycolysis? We produced 4 ATP, right? What is 4 ATP here? Sorry, a little slow on my writing there. Okay, well, guess what? We're going to need those 4 ATP to reconvert into glucose. Okay, so it is going to be an endothermic reaction overall. We are going to have to input energy in order to build this molecule back. Okay, so glycolysis breakdown, we release energy, glucogenesis build up, we need energy to reconnect those bonds. Okay, so steps are pretty much the same. Okay, we are going to add carbon dioxide a little bit. So we're going to go pyruvate to oxaloacetate. You remember oxaloacetate, we're talking about Krebs cycle here in the mitochondria. So I'm actually going to add some CO2 here. Okay, and then we're going to input some energy. Okay, and again, every step requires an enzyme. Okay, but if you notice, most of these enzymes are the same enzyme. Okay, so they are reversible depending on the circumstances. Okay, so part of that is where are we at? Okay, so we're going to get more glycolysis again in the muscles. We're going to get more glucogenesis in the liver. Okay, and so the one step that we're going to focus on here is this step from fructose 1,6-biphosphate, okay, to glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. Okay, and again, that enzyme that's going to catalyze that is aldolase in both sides of the reaction. All right, so effects of aldolase. Okay, it's going to catalyze that reversible step. So about step number 4 in the process of glycolysis, okay, which is breaking down again the 6-carbon fructose 1,6-biphosphate into its 2 3-carbon halves, essentially, if you want to keep it simple. Okay, so in glycolysis, it's going to break it down. In glucogenesis, it's going to do exactly the opposite and build it up. Okay, so if we look at the picture, okay, so this is a reversible reaction with aldolase. Okay, essentially, it's going to cleave this 6-carbon molecule that you can see here into basically halves. Okay, so slightly different molecules, but not very much, and these can be converted. Okay, again, dihydroxyacetone can be converted into glyceraldehyde 3-phosphate. Okay, so aldolase plays that key step. Without it, none of this works. Okay. All right, so now we get into our different forms of aldolase. Okay, so aldolase, because it does both jobs, okay, we're going to see 3 types of aldolase. Okay, aldolase A is the most common one that we'll see in muscles and red blood cells, and that's going to basically only function in glycolysis. So it's going to break down sugars into pyruvate. Okay, and as you see, pyruvate will either become, there's pyruvate, right? Pyruvate will either become lactate or it will go through the TCA or citric acid cycle or a Krebs cycle and become carbon dioxide at the end of it, okay, through the electron transport chain. Okay, so the end result here would be CO2. Okay. All right. Aldolase B, okay, is going to be primarily found in the liver. Okay, so this one's going to be found in the liver. Okay, and it's going to act in both glycolysis and gluconeogenesis. So this one's going to be where it functions both. So one of the big ones is it's going to break down fructose. Okay, and fructose being another form of sugar. Okay. But it's also going to reconvert things like lactate. So lactate would roll over here, okay, and in the presence of lactate, aldolase B is now going to, instead of breaking down glucose, it's going to reconvert that lactate into glucose. Right? And then we'll go through the process again. So it seems kind of backwards there, but you'd think, okay, we can just convert lactate directly back into pyruvate. You know, it does require a reversal into glucose, okay? One, because it needs to be transported to each and every cell, okay? And glucose obviously being a very stable molecule. So no, we don't convert directly back. We convert back into glucose. Glucose is then moved throughout the blood to the body, okay? So this is fructolysis, okay? The breakdown of fructose, okay, and the last one is aldolase C. Aldolase C we're going to see primarily in the brain. Okay, if you recall your physiology on the brain primarily works only on carbohydrates, only on sugars. Okay, and that's to remove a lot of the waste products that might be produced in the process. And so aldolase B, or aldolase C, excuse me, is going to provide our energy for our neural tissues, okay, and again, mostly in glycolysis, okay, so anyway, so A and C, glycolysis, B does both, but more involved in the glucogenesis process, okay? So a little bit slightly different molecules, okay, so regulation. How do we regulate aldolase B? Okay, so we got 4 key transcription factors, which of course are affecting the DNA. Okay, and essentially we're controlling blood sugar here. Okay, so we're controlling both glycolysis and glucogenesis. Okay, so part of this is again going to come back to our hormones. Both insulin and glucagon are going to affect, okay, which way we're going to go with this, glycolysis or glucogenesis, okay? And with all of the enzymes essentially being reversible, okay, it's now a matter of what do we have? Do we have a lot of lactate? Do we have a lot of glycerol and not a lot of glucose? Then we're going to go through glucogenesis. If we got a lot of glucose, a lot of sugar in the body, then we're going to go the opposite way. We're going to go through glycolysis and break down that sugar. Okay, so remember, a lot of it has to do with the levels of the material. Okay, so key transcription. How do we essentially make more aldolase? Okay, so we're going to have two key ones, HNF1, HNF3. Okay, so these two here on top, okay, go and work together. Okay, so it's actually the 3, HNF3, hepatocyte nuclease factor 3. Okay, that one's going to actually be a broader aspect. It's going to essentially break open the chromatin structure of the DNA. So it's going to separate it into 2 strands, or it's going to break the ionic or hydrogen bonds between the DNA molecules, okay, between the opposite base pairs. You know the drill. Okay, and it's going to open up that to more binding. And since it's open, the RNA polymerase and other factors will bind more readily to it, and we're going to get more transcription. So that's going to be where we start. HNF1 is going to actually activate the promoter region. Okay, so it's going to bind to and activate the promoter of the aldolase B gene, okay, which is going to enhance the transcription, which will in turn enhance the translation. Okay, so most of our control, again, is going to happen in transcription. Okay, but even though we do copy it, there is the ability to control at the translation point, okay, based on levels of material as well. Okay, so the next one we got here is CCAT, or enhancer binding protein. This is going to activate more of the genes involved in glucogenesis. Okay, so CEBP, okay, is going to focus more on glucogenesis, okay, and rather than glycolysis. And lastly, we have D-site binding protein, or DBP, okay, and that one's going to work more on our circadian rhythm and just regulate it throughout the day. So let's take a look. Okay, so again, we got aldolase B, okay, which is our primary one. Actually, let's go to the next slide. There we go. All right, boom. Okay, so HNF1 we see here, but HNF1 is going to be contributed with multiple other ones. HNF3, HNF4, okay, they're going to stimulate or break open that DNA, and then HNF1 is going to produce multiple different genes. Okay, aldolase B is only one of those. Okay, because obviously it's not the only enzyme in the process. Okay, but they're going to directly control it. Okay, on the other end, okay, a separate role is C/EBP alpha. Okay, and that one's going to be responsible mostly for glucogenesis. Okay, so that's going to be aldolase B again. Okay, but if you notice some of the other ones that are produced, some of them are the same, albumin, okay, AFP, okay, PEC, TAT, okay, but notice there's a couple different ones here, okay, which are going to, again, push it towards glucogenesis rather than glycolysis. And again, everything is regulated, again, by what is there. Okay, so what does the body need? Is the blood sugar too high, blood sugar too low? Okay, that's going to affect which way we're going to go. More often than not, we're doing glycolysis. Okay, glycolysis, again, will lead us to cellular respiration or oxidative respiration. Okay, in anaerobic conditions, okay, we are producing lactate. Okay, and following the buildup of lactate, obviously, we'd go through glucogenesis. Okay, so wrap it all up. Glycolysis, breakdown, glucogenesis, buildup of glucose. Okay, aldolase, key step, which breaks down their 6-carbon molecule, okay, in this case, fructose 1,6-biphosphate, into its 2 3-carbon pieces, okay, and that's going to be a key step in activating the rest of the process, okay, and again, it is reversible. So remember that the big combination between these two is the reversibility of most of the enzymes. Okay, so you think about it, normally when we break something down, can we really build it back up? Well, yeah, the pieces are still there. What does it need? Needs energy. So glycolysis, release of energy, glucogenesis, we need energy, but we're going to get more out of that glucose molecule. So put in a little, get a lot out later. Okay, hope you enjoyed the presentation. Thank you.